IB Biotranscription Translation Part 3 will focus on the process of translation, where a code in the mRNA is translated to a sequence of amino acids in a polypeptide. The essential ideas are genetic information in DNA can be accurately copied and can be translated to make the proteins needed by the cell. And secondly, information transferred from DNA to mRNA is translated into an amino acid sequence. Before translation happens in the cytoplasm, the raw mRNA transcript is processed. Introns are removed, exons are spliced together. As well, the transcript is modified with the addition of a poly A tail and a 5' guanine cap. In this diagram, the mRNA exits the nucleus and becomes bound to a ribosome. A ribosome is composed of two subunits and has multiple binding sites, including one for the mRNA. The ribosome is a large enzyme that synthesizes a polypeptide using the RNA code as the template to determine the amino acid sequence in the polypeptide. In this diagram, we see the 2-unit ribosome bound to the mRNA and moving along the mRNA in a 5-to-3 direction. The ribosome serves to synthesize the polypeptide one amino acid at a time. So here is the first IB syllabus statement of relevance. Outline the structure of ribosomes, including protein and RNA composition, large and small subunits, 3 tRNA binding sites, and an mRNA binding site. Ribosomes are composed of both RNA, ribosomal RNA, and protein. The 2 subunits can be seen here, a large subunit and a small subunit. The ribosome has multiple binding sites. The ribosome binds the mRNA and has 3 binding sites for transfer RNA molecules, the A site, the P site, and the E site. We will learn more about transfer RNA and the 3 binding sites on the ribosome later in this movie. DNA, located within the nucleolus of the cell, has the genes for ribosomal RNA. The genes for ribosomal RNA are transcribed in the nucleolus. Numerous RNA molecules aggregate together along with protein to form the subunits of a ribosome. In this diagram, we can see various ribosomal RNA molecules formed within the nucleolus, and these rRNA molecules will aggregate together along with protein, which arrives from the cytoplasm, to become the subunits of ribosomes that leave the nucleus to the cytoplasm, where they function as the full ribosome. Ribosome size and character can be used to distinguish among evolutionary lines. The eukaryotic cells have 80 S ribosomes, while prokaryotic cells have 70 S ribosomes. Here is a good diagram that displays the 2 subunits of a ribosome and the 3 binding sites for tRNA, as well as the binding site for mRNA. Here is an important IB syllabus statement. Explain the process of translation resulting in a polypeptide with an amino acid sequence determined by the mRNA according to the genetic code. This is for HL and SL students together. In this diagram, the mature mRNA molecule exits the nucleus and becomes bound to the ribosome that translates the nucleotide base sequence in the mRNA into an amino acid sequence of the polypeptide. The function of the protein depends on whether the ribosome is free in the cytoplasm, as it appears in this image, or bound to the endoplasmic reticulum. In this diagram, the mRNA leaves the nucleus and becomes bound to a ribosome that is located on the ER. The polypeptide is then deposited into the ER, where it is modified and then transported to the Golgi for further modification. From the Golgi, the functional protein is ready for secretion from the cell in a Golgi vesicle. The protein might be secreted into the bloodstream if the protein is a hormone, or it could be secreted into a duct if the protein is a digestive enzyme. In this diagram, we see ribosomes in contact with the endoplasmic reticulum. The newly synthesized protein is deposited into the lumen of the ER for modification and transport to the Golgi. Following further modification in the Golgi, the protein will be moved into a Golgi vesicle. The Golgi vesicle could become a lysosome for use within the cell, or it could be used to transport the protein to the cell membrane for exocytosis, secretion out of the cell. So here's an IB syllabus statement that you should already know. State that free ribosomes synthesize proteins for use primarily within the cell itself, and that bound ribosomes synthesize proteins primarily for secretion and lysosomes. Here is a micrograph of rough ER studded with ribosomes synthesizing protein for one of two fates: lysosome or secretion out of the cell. In this micrograph, you should be able to see both ribosomes in contact with the rough ER, as well as free ribosomes. Free ribosomes synthesize protein for use within the cell, such as for enzymes, cytoskeleton, microtubules, etc. Here is a diagram of many ribosomes simultaneously translating the mRNA in a 5-to-3 direction. These ribosomes are free in the cytoplasm. Numerous ribosomes simultaneously translating the mRNA is known as a polyribosome, or polysome for short. Here is a micrograph showing numerous ribosomes bound to the mRNA simultaneously translating the mRNA. This is a polyribosome or polysome. The ribosomes are synthesizing polypeptides for use within the cell, such as for enzymes or microtubules, the spindle fibers, for example. So here are two relevant IB syllabus statements. Can you identify polysomes in electron micrographs of prokaryotic cells and eukaryotic cells? And do you know that translation can occur immediately after transcription in prokaryotic cells because of the absence of a nuclear membrane? Here is another micrograph showing numerous ribosomes translating the mRNA. The newly formed polypeptides can be seen here. The polypeptides will be used within the cell. This entire structure is known as a polysome, numerous ribosomes simultaneously translating the mRNA. Remember that just as transcription happens in the prokaryotic cell, ribosomes can bind to the mRNA and immediately begin the process of translation. Now here's an IB syllabus statement with which you should be very familiar. Describe the genetic code in terms of codons composed of triplets of bases. Remember, the mRNA nucleotide code is a triplet code. In other words, 3 bases as a triplet code for 1 amino acid. Here you can see the mRNA nucleotide sequence, but translation occurs in triplets where 3 bases as a codon result in 1 amino acid in the polypeptide. So here is the codon dictionary, something that scientists have known for a long time. We can see where a codon like GGG codes for the amino acid glycine, or UUU codes for the amino acid phenylalanine. These two IB syllabus statements will carry us into the next movie. Explain the process of translation resulting in a polypeptide with an amino acid sequence determined by the mRNA according to the genetic code. Describe the genetic code in terms of codons composed of triplets of bases that correspond to 1 amino acid in a polypeptide. Here is a nice diagram that displays the triplet code. Triplets of mRNA nucleotides are translated by the ribosome. Each triplet codes for 1 amino acid, as you can see here. So the question is, what links the mRNA nucleotide sequence, where I have the codons color-coded here in triplets, to the formation of peptide bonds between the specific amino acids called for by the triplet codons? In other words, what serves to translate the nucleotide sequence into the amino acid sequence? And the answer is transfer RNA. In this diagram, we can see the mRNA, we can see the ribosome, and for the first time among the images I've presented, we can see the tRNA molecules transferring the appropriate amino acids into position in the growing polypeptide chain. At this point, I'm providing a general view of translation. The details will follow in the next movie. One more look at the overview of translation. The mRNA carries the code, and the tRNA translates the code by transporting the appropriate amino acid to the ribosome. And the ribosome is the site where peptide bonds are formed between the amino acids in the synthesis of the polypeptide. As one last idea, it's important to note that the genetic code is universal among all organisms. The universality of the code has allowed humans to transfer the human gene for insulin into bacteria, and bacteria will synthesize insulin. This illustrates that the genetic code is universal. The gene for insulin is withdrawn from a human cell and inserted into a ring of DNA known as a plasmid. The plasmid is a naturally occurring ring of DNA found in prokaryotic cells. The plasmid with the human gene for insulin is then inserted into bacteria. The bacteria synthesize insulin. Human insulin is synthesized by bacteria. Type 1 diabetics who need insulin rely on bacterial factories to synthesize insulin from the human gene inserted using a plasmid. And that brings us to the end of IB Bio Transcription Translation Part 3. Part 4 continues with higher level material on translation.